Hi guys, so we are doing the last set of notes for DNA. To make sure that at this point you go back, if you missed any of them, I will be grading notes pretty soon. So how it works with the virtual is if you hadn't noticed, is I go back and I check each set of notes from this unit and I average together your grade. So if you only did one of the sets of notes, even if you made 100 on that set, well, there were four or five parts. I can't remember now. I'm gonna have to go back and look. But if you know you've missed some of them, like you're automatically gonna get average in those zeros of you just not doing them. So the note grade gets pretty low. All right, so this is where we left off. So let's move forward. So steps of DNA fingerprinting. So we went over this on Friday. Y'all did the virtual lab for it just to kind of learn by doing, seeing what the process is. Now you'll have it in your notes. So steps of DNA fingerprinting, how we actually make those set of bands we can match to our suspects or match for paternity is first, obviously, is extract the DNA. We're actually going to have an extraction of DNA lab tomorrow. Yeah, tomorrow. So on Tuesday. But for a person to extract the DNA is most of the time they just do a cheek swab. You can get it from blood and all those other places we've talked about where you can get DNA. But if we were like collecting a sample from a suspected test, it's usually a cheek swab like this. So they're trying to get some cheek cells. Step 2, cutting the DNA into restriction fragments. So step 2 is we mix that DNA with restriction fragment enzymes. Those restriction fragment enzymes are also called restriction fragment length polymorphisms. I'm not going to say all that. I'll usually just say restriction fragments. Okay, so what they're going to do is they find a restriction of basically a code they're searching for. So like an example, let's say a restriction fragment enzyme is looking for the code AATT. So they put that in with a DNA, and everywhere in that person's DNA that they find AATT, they're going to stop, make a cut. And that's going to be the length of that fragment. However many base pairs, so AATT, however many they passed before they found AATT, the code they're looking for, that's how big the piece is that they cut. Now, the next piece they cut, they might find AATT faster. So it might be a smaller piece that they are restricting and making a fragment length of. And then it might be a really, 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 really long piece before they find AATT again. But wherever they find it is where they're going to make the cut. They're going to put that same restriction fragment enzyme in the suspect's DNA as the crime scene DNA. If they are the same person, if the crime scene DNA is the same suspect, well, then the restriction fragment enzyme should be making the same size cuts. Step 3, we take those pieces. So now your DNA is cut into pieces and it goes into amplification, making many copies of those fragments PCR. So like I said before, we don't want to ever run out of our crime scene sample, our DNA sample. So they're going to do PCR pretty much always because we want to make sure that we have enough to save, be able to run more tests as needed. And sometimes you just need to run PCR because the sample size was so small. The DNA itself, like just that little sample you got, wasn't very big. So just to make it enough to run through electrophoresis, they're going to amplify it, meaning they're going to do PCR, which is making copies. And then lastly, step 4 is electrophoresis. So that is that machine that you should have saw in the video opener on Friday. And that's the machine that you worked through on the virtual lab. It's basically a little box. It has a negative end and a positive end. It has little holes on one side. That's where the DNA goes into those holes, these little wells in a gel. They turn the machine on. Remember, DNA has a negative charge. So over here it's negative, over here it's positive. It wants to go towards the positive end because it's attracted to it. So it starts moving across. Remember how it moves across is you have those pieces of DNA, and some of them are big pieces, some of them are little pieces, and the little pieces make it further than the big pieces that don't make it as far, which ends up being those DNA bands that we've been looking at, like this. So you put the DNA here at the top. They're trying to make it down here towards the bottom. The littler the pieces are, the further they make it through the gel in the time period that the machine is turned on. The bigger pieces don't make it as far. One should contain a control. So if you notice, like these look really evenly spaced, it's because this is a control. Let's put in this as a sizing marker or a DNA ladder, as the video called it on Friday. A lot of things we called known, called marker, standard DNA, DNA ladder, all those are this meaning the same thing. It's just a control. And the control is put in there to show sizing. So like this is, I know how big those pieces are. Like maybe we'll say this one's 500 links long. This is 100 links long. This is 1,000. Usually it's like, you would write it down off to the side. Like this is our known marker and we know that they're this size. So if this is that size, then I can determine my unknown sizes because it's lining up with my control sample. Other wells should contain the DNA from the crime scene, the victim, and the suspects, as you're seeing here. So you are some suspects, you have the victim, you're trying to see which one they match. Analysis of DNA fingerprints. DNA fingerprints look like a striped column. In order for the DNA fingerprints to match, the columns must have bands in the exact same places with the exact same width. So they do have to be exactly the same or they are not a match. How gel electrophoresis works. This was in the video. I just talked about it, so I'm not going to spend a long time, but you should have this picture here. You can even watch videos. I'll post this at the top of the week, this PowerPoint, if you want to see it again. So the restriction enzyme. So first you have your DNA sample. Take it, cheek swab. We mix it with your extraction enzymes, and they cut the DNA into different size pieces based on where they find that code. Example, finding AATT. There's lots of codes that you have restriction enzymes looking for, but you pick one, that's what it's looking for. The DNA segments are loaded into the wells in a porous gel. The gel floats in a buffer solution. So there's gel and there's like some liquid solution around it. When it is turned on, there's an electric current. And where the DNA is put in, see this negative symbol? It's negatively charged here. Well, DNA is negative because phosphates in DNA are negative. The phosphate sugars. Phosphates are negative, so DNA has an overall negative charge. If I turn this machine on and it's negative right next to where the DNA is, what happens when I have two negative things? Like I have two negative magnets. They repel. So just by turning this on, it is repelling those DNA pieces to move away from it because it's like, I don't like that because same charges repel each other. It also is being pulled to this side that's positive because opposites attract. So this DNA is negative and it's moving towards that positive charge because it's attracted to it. Okay, so you turn the machine on, the little pieces start doing their moving. These smaller DNA segments move faster because they're lower in weight. They're lower in molecular weight, so they can move faster through the gel, so they make it further down here. And the bigger pieces are slower because they are heavier. They have a heavier molecular weight. They just can't make it through the gel as fast. So they end up being up here, making our DNA bands that we can then match to our suspect. Kind of skip this. DNA fingerprinting can match crime scene DNA with a suspect, eliminate a suspect, free a falsely imprisoned individual because we realized they didn't match and we had accused them, determine maternity, paternity, or match to another relative. So if that's the one way to figure out who our Jane Doe is matching to their parents to see if that's their DNA, that could be a way to identify a missing person. Could be a way to figure out just plain paternity, like is he the father? All these are done with these DNA bands, and identifying human remains is what I just talked about. The DNA profile databases. So in every unit, I've talked about like the main database for that kind of evidence. Each state keeps DNA profiles of individuals who have been convicted of certain kinds of crimes, such as rape, murder, and child abuse. Every state has theirs on file. The military maintains DNA profiles of all the servicemen and women. So if you go into the military, they have your DNA. The United States electronic database of DNA profiles is called CODIS, Combined DNA Index System. So the big database where we keep all of our main DNA cases would be CODIS. All of the jobs that require it, like the military, would be in CODIS. As of February 2011, CODIS contained 9.4 million profiles. So this was 9 years ago. I need to look up a current one. That number is going to have gone up. From the DNA from convicted felons or violent crimes, of 360,000 profiles from the DNA to crime scenes, CODIS made 138,700,000 hits. I can't say the word, 138,700 hits. There we go. Just showing you that, like, obviously having it in our database helps us catch bad guys. If we could get more into the system, we probably could catch more bad guys. So that's kind of like the little philosophical debate that I like to ask kids at this point. Should we have more of us in the system? Is that against our rights to have to put our DNA into a system, or is it just helpful for society? Because then we would just have us all on record. Like we just prick little babies' feet as soon as they come out. Here you are. If you ever become a bad guy, we'll know. I don't know. We definitely would be able to stop more people from doing these terrible things. I don't know what it could be used bad for. There's a lot of like, what do you call it? Always forget the word when people think like the government's going to use it for something bad. Never mind. Sorry. Making notes take longer and I can't remember the word, but ponder that. Do you think it's something that we should have more jobs that require us to have it? Put our DNA on file or not?